This program is a presentation of UCTV for educational and non-commercial use only.
It's a pleasure to be here tonight. I changed the title of my talk. It originally was Worms, Flies, and Yeast, and I thought that didn't sound very, people probably just had dinner and that just doesn't sound good. So I changed it to Breadfruit and Compost. And hopefully it'll become clear to you as the talk goes along why that is the title. So Bruce asked Keith and I to talk a little bit about the frontiers of research in model organisms. And so we sort of divided up the model organisms, and I'll be talking about the spineless ones. Okay. So as an overview to what I'll be going over tonight, first I'm going to talk a little bit about how physicians and scientists think about studying neurodegenerative disease as an introduction. And then I'm going to talk about what actually are model organisms and why are they useful. And then I'm going to go from there and actually talk about a couple of different areas that are really sort of on the cusp of the cutting edge of research in neurodegeneration today, using a couple of examples, some from my own work and some from others. And we'll be talking in particular about FTLD and ALS, and then Keith will be talking a lot more about Alzheimer's disease. Okay. So first, how do physicians and scientists study neurodegenerative disease? Okay. So this seems like sort of a philosophical question, but I think it's really an important one, and it sort of forms the backbone of the talk I'm giving tonight. And I'm borrowing a schematic from my friend Charlotte Sumner, and I've adapted it somewhat. And this is now how I think about how do you organize a study into a condition. So, for example, I still see patients in clinic at the Memory and Aging Center, but for example, what if someone presented to me with something new? Let's call it blue face syndrome, okay? No one has ever heard of this before, but now someone has it. And how do we study it? How do we understand what causes it? What are the risk factors? How can we treat it? And how can we prevent it from happening again? Okay, so using blue face syndrome as an example, first an individual actually has to present with the condition, okay? So Mr. X comes to me in clinic, has a blue face, and I want to figure out what's going on. I don't know. But let's say next week someone else presents with blue face. And so now, I find out they both live, in San Francisco, maybe in the Richmond district. And so I'm going to describe it. I'm going to put a paper or, write a Twitter feed about this blue face syndrome I've discovered in San Francisco in the Richmond district. And then once I've done that, then other people can then say, oh, look, last week I saw someone with blue face syndrome. And once that propagates, then a cohort or cohorts can be organized, and then we can actually understand something about, I've written here genes, but maybe the risk factors that cause it. Maybe living in the Richmond is actually a risk factor for blue face syndrome. And so once we do that, then we can start interrogating, well, not only do those people live in the Richmond, but maybe they all had sushi, last week. And so maybe the risk factor is eating at a sushi restaurant in the Richmond. And so now we understand something about the disease, and we can understand something about the mechanism. We can start thinking about targets of therapy. So, maybe there was a microorganism in the sushi, and so we can figure out what that microorganism is and then treat it, okay? And if we're really lucky, or mostly lucky, but maybe if we work really hard, we can also maybe prevent the disease from happening in the first place, okay? So that's sort of a fanciful example, but we're going to go through this schematic the rest of the evening, okay? So this is, in reality, how people have approached the study of many diseases, including neurological diseases like epilepsy, migraine, multiple sclerosis. cardiovascular diseases like diabetes and heart disease, psychiatric diseases like depression, schizophrenia, mental retardation, and some that's not even a disease, but aging, okay, which I actually I have trained in Cynthia Kenyon's lab here at UCSF, who sort of started the study of aging, but we can think about aging even in this way, okay? But tonight, we're not going to talk about those diseases. We're going to talk about, or at least me, frontotemporal lobar degeneration and a closely related condition called ALS. Okay. So you all already heard last week from Dr. Seeley, I believe, a lot about FTLD, or frontotemporal lobar degeneration. So I'm not going to go into detail about the disease since you already know quite a bit. But I'm just going to go over sort of the briefest, barest bones of it. And that is historic. We'll go back in time to 1892 when this gentleman, Arnold Pick, had some patients present to him with some unusual symptoms. And he wrote a paper that described what came to be known as Pick's disease, which is a subtype of FTLD, which I believe Bill Seeley went over last week. The disease was found to preferentially affect the frontal and temporal lobes. And later on, not by Arnold Pick but others, found that a subset of cases are associated with ALS, which is also known as Lou Gehrig's disease. And so there's probably something about these two diseases in terms of how they develop, what causes them, that are similar. And finally, a large percentage, about 40% of cases are familial, meaning more than one person is affected in the family. And this is, of course, very, very sad. It's tragic for the families. But from trying to figure out what's going on, from that point of view, it's actually a boon to scientists because we're able to use those families to try and narrow down what's actually the causative factor. And in families, usually it's a gene or a change in a gene. Okay, so I just gave you some information that could sort of fill in these three boxes. First, a patient or patients with frontal lobe symptoms presented to Arnold Pick. And then he described what came to be known as Pick's disease. And then more individuals with Pick's disease were identified. So now we want to find the cohorts. So in the late 1990s, Maria Spillantini and her colleagues actually identified a family that had frontotemporal lobar degeneration. And in case you've never seen one of these diagrams before, these are several generations, actually 7 generations of members of a family. This is the index case right here. I think males are in the squares and females are the circles. And all the individuals where the box or circle is filled in with black were affected with the symptoms, okay? And then the individuals with a little cross, like these, have passed away. And then the individuals that have the little dots under them actually had their DNA studied by these scientists. And because there were so many people here that both had the syndrome and didn't have the syndrome, these scientists were able to use what's called genetic linkage studies or genetic mapping to actually narrow down and actually find the one gene that was changed in all the people that had the disease and it was not changed in the individuals who didn't have the disease. And in this family, the gene that they identified was known as tau. Also, and the gene name is also MAPT, for microtubule-associated protein tau. I'm not going to be talking a whole lot more about tau. Keith will be talking more about tau. But in quick succession, well, not so quick, but over the intervening years, several other genes have been associated in a very similar way using this genetic linkage to identify genes that can also cause frontotemporal lobar degeneration. And these include progranulin, VCP, something called CHIMP2B, FUS and ALS. Oh, excuse me, FUS and TDP43. And these last two genes actually have only so far been associated with the development of ALS. But there's a good chance that families or individuals will be found that have mutations in these genes that may also present with frontotemporal lobar degeneration. So I am not going to talk about actually VCP, CHIMP2B, or FUS anymore, but I'm going to spend some time talking about progranulin and also about TDP43. Okay, I want to take a little sidestep and just tell you this interesting story, though. Okay, so I mentioned that this gene, MAPT or tau, was the first gene that was identified that was shown to cause FTLD. okay? And scientists did this by doing this sort of narrowing. First, they found the chromosome that the gene was on with these linkage studies, and eventually they narrowed it down to tau, or MAPT. There were several other families that existed in the world, and scientists were sort of busily mapping also where the mutation was in those families. And as soon as this MAPT or tau was published, several other groups in the world said, oh, well, our families also have linkage to chromosome 17, and so probably what they have are mutations in tau. So they set about sequencing the tau gene forwards, sequencing it backwards, sequencing the introns, sequencing the exons, sequencing around it, because they couldn't find a problem actually in the gene. They even looked to see were there duplications or triplications in the gene, because that has actually been shown with alpha-synuclein in Parkinson's disease. And they just couldn't find anything wrong with tau. And so finally, people began to look. So it was this red herring, basically. Turns out very close to tau was the gene for progranulin. And it's so close that, I mean, really, all you can say about it is it's just an extraordinary coincidence. Now, as it turns out, this piece of DNA right here is actually very, very old. This little piece was sort of conserved from the Neanderthals on downwards. So there may be a reason why these two genes cause this very similar disease that are so close on this chromosome, but we don't know. But as one scientist who studies FTLD said, finding these two genes right next to each other, I mean, it's extraordinary. It's like a 1 in 100,000 chance that these two genes would be right next to each other. It's like finding a needle in a haystack next to another needle in a haystack. I mean, that's sort of the analogy that has been made. So it's really an extraordinary coincidence. Okay, but let's get back. We found our cohorts and we found the genes. Okay, so the families with Pick's disease and FTLD were identified, and then tau, progranulin, TDP, et cetera, were found. So now we want to know, well, what can these genes tell us about the mechanism of disease? Because that's how we get over here to targets and treatments. Okay. So most scientists today would use model organisms to try and get at these mechanisms. And in order to tell you why, let me tell you a little bit more about model organisms. Okay, so what are model organisms and why are they useful? Okay, so this is the phylogenetic tree of life. And here we are, all of us, up at the very top, vertebrates. Okay? And down here, way below us, are things like amoebas and slime molds and, you know, algae and flagellates. And these are all, in and of themselves, fascinating creatures and could be very good model organisms. But I'm not going to talk about those. I'm going to talk about 3 model organisms tonight. And they are yeast, the baker's yeast, Saccharomyces cerevisiae, which besides being used in hundreds of labs every day around the world, is also used every day in hundreds of thousands of bakeries to make bread. The roundworm, the tiny microscopic roundworm, Cenorhabditis elegans, which is shown here. It looks scary, but it's only 1 millimeter big. And it's very important. The C. elegans is very important to us because now, living in San Francisco, we are all composters. And C. elegans is actually the worm, not the earthworm. It's C. elegans that helps to make our rotten fruit and vegetables into compost. And finally, the fruit fly, Drosophila melanogaster. And I like this picture, but it's an artist's rendition because these little fruit flies are not actually the size of golf balls. They're actually the size of ants. So this is either an enlarged fruit fly or a very tiny banana. Okay, so those are our topics for my topics for the rest of the evening. So why use model organisms? And my subtitle for this this slide is I like to think of it in my mind as no, you can't clone my grandmother. Okay, because there are ethical considerations in human research, and that is right and good that we think this way. Do unto others as we would want done unto us, and I don't want anyone doing research on me. And so I'm not going to do it on anyone else unless I know that it's at least safe. Okay, so that's I think the #1 reason we use model organisms. But there are additional reasons, and this includes that the basic biological processes are the same. We are eukaryotic organisms. A yeast is a eukaryotic organism, meaning our cells have nuclei, they have mitochondria, they have endoplasmic reticulum, we synthesize proteins. All those processes are the same, and if we're studying those basic processes, we can do those in model organisms. in model organisms, we can much better control the genetics and the environment. And if we're looking for small changes, we want to remove the noise from our systems. And differences in genes and differences in environment, for example, you could have two twins. Let's say they're genetically identical. Well, maybe one had a hamburger and another one had a hot dog for lunch. There, we've introduced an artifact into our studies. So we can control genetics and environment much better with model organisms. Okay. We can also amplify and repeat studies. In the lab, I could do an experiment tomorrow on 1 million worms. And then I could do that study again next week. And that's very, very powerful to be able to harness those numbers and to do studies more than once for a reasonable cost, I guess. And there are wonderful tools and techniques, some of which I'll tell you about, available. And finally, it's just because they work. Every one of us in this room has probably had a vaccine or taken an antibiotic or had a rash and treated it with a cream. And all those studies to make sure that those treatments were safe were all done in model organisms. Okay? So there's a long history to show that these types of approaches work. Okay, but that's one thing. A rash, being able to treat an infection, that's one thing. We're talking about neurodegenerative disease. And is it really realistic? Can a tiny, spineless, brainless creature teach us about neurodegenerative disease? And I should say that it's not actually true that flies actually do have brains, and worms have something close to a brain, but yeast don't have brains. But I just want to be accurate. So, but is it true that these things could actually teach us about neurodegeneration? I think it's a very reasonable question. And so in order to try and answer that question, I'm going to take my favorite model organism, although I like them all, which is C. elegans, and try and illustrate how important C. elegans can be in scientific research. So here's a better picture of C. elegans. Okay? C. elegans was plucked from obscurity by a Nobel Prize-winning scientist named Sidney Brenner. He literally was sitting in his study one day thinking, we need a new model organism that we can do genetics on. And so he went out to his backyard in Bristol, England, and he dug around in his compost heap, and he found these little worms, okay? And these worms are fabulous. You can tell I'm a big fan. They are small. They're 1 millimeter big. Like I said, I could grow 1 million of them later this week, okay? They're really easy to maintain. We just keep them in little incubators on plates. They have a very simple anatomy, but we understand it all. And there are analogies to human anatomy, okay? Here's the head. Here's the tail. We don't have tails, but there's an intestine that runs through the body. There's skin. It's called a cuticle. There's muscle. We can't see it, but the muscles are very similar to human muscles. There are reproductive organs, and there are neurons. There's a nervous system, okay? They're genetically tractable, which means that we can do things to manipulate their genes. We can, well, number one, they have two sexes, just like humans, but their sexes are male and hermaphrodite, which is a self-fertilizing animal, okay? And so you can grow these hermaphrodites in the millions, and they would be isogenic. They'd be identical genetically to one another, okay? And then we can knock down genes, we can delete genes, we can overexpress genes, we can express mutated forms of genes, we can express human genes in these animals. And so they're very manipulatable from a genetic point of view. They're also transparent, which is huge because a lot of the work that I do is microscopy, and to be able to, for example, our skull, you can't see through it, but these guys have neurons, and we can actually see them in a living organism. We don't have to fix the tissue to look at it. We can actually look at what's happening while it's happening. And finally, they have lots of complex behaviors, which I'm going to get to a little bit more in a second. Okay? And to illustrate how important C. elegans has already been in the study of biology and human diseases, I just have a few examples. So there's something called programmed cell death, which is well, I'll tell you a little bit more about it in a little bit, but basically it's how cells decide to die, okay? The study of programmed cell death was really pioneered, and the basic molecules involved were all described in C. elegans. There's something that you might have heard of called RNA interference, or RNAi, which is becoming increasingly important in therapeutics for human diseases. And although the initial observations were made in Arabidopsis, a plant, once again, the molecules were worked out in C. elegans. There's something called green fluorescent protein, which you may have heard of. It's this glowing protein that allows us to see things in cells in real time, and that work was first done in C. elegans. Here at UCSF, as Bruce mentioned, the study of aging has been pioneered by Cynthia Kenyon, and it's since been shown that many of the genes and molecules that influence aging in worms also hold true in at least apes. And there's some indications that the similar genes are important in humans. And finally, Gil Rabinovich, you might have mentioned presenilin. It's one of the genes that can be mutated in familial or genetic forms of Alzheimer's disease. And presenilin was actually first sequenced and identified in C. elegans. And to sort of an external reviewer would say, well, how important really are these? Well, they look like pennies, but this is actually Alfred Nobel. So Nobel Prizes have been awarded for work in these three areas to people who worked primarily in C. elegans. So C. elegans is actually a very, very important organism. But I told you it only has sort of a brain, okay? So here's a human brain, 10 to the 10th neurons. Here's C. elegans with its neurons lit up with this green fluorescent protein, and it has 302 neurons, okay? So I think it's fair to say that 10 to the 10th and 302, there's a big gap in there. But that being said, these 302 neurons are extremely well described, and they actually have the same neurotransmitters as humans, okay? So dopamine, acetylcholine, GABA, glutamate, norepinephrine, serotonin, they're all in worms, okay, represented in worms. And like I mentioned before, there are loads of behaviors that we can study to try and see if we do something, if we put in a gene, is there behavior that changes? And these behaviors include coordination, or how the animals move, reproduction, chemotaxis, or how these animals sense and respond to the environment, and even learning and memory. So last year there was a paper published by a former postdoc here at UCSF who showed that you can actually teach worm things, and then you can measure if they remember them. And it's very robust. And so now we can even study learning and memory in worms. Okay? Did you know that, Bruce? That's pretty neat, huh? Really neat. Okay. So I've told you about model organisms, why they're useful, and now I'm going to get into a little bit of the frontiers in FTLD research, and this is the first story from worms to mice. Okay, so I mentioned this gene, progranulin, that can cause frontotemporal lobar degeneration in humans. And this is a model of the gene, okay? And C. elegans has progranulin as well, and is oftentimes the case. It's a little bit simpler than the human version. And being a simple person, I actually like that. Neurodegeneration is a complex thing. If we can simplify it, let's simplify it, okay? So the question is, what does progranulin do? Okay, and how does not having enough cause neurodegeneration? Well, there are a couple things known about progranulin when it was identified in 2006. One is, well, in 2006 it was shown that if you don't have enough progranulin, you get neurodegenerative disease. But as it turns out, if you have too much progranulin, that's bad too, because tumors, particularly aggressive tumors that like to metastasize, produce loads of progranulin, and these include cancers such as breast cancer, a brain tumor known as glioblastoma multiforme, kidney tumors, bladder tumors, lots of tumors that are particularly aggressive produce lots of progranulin. And so people thought, when these two things were found, that, well, progranulin, it must be a growth factor, right? Because if you don't have enough of a growth factor, you get neurons dying, you get neurodegeneration. If you have too much of this growth factor, you cause cells to grow uncontrollably, and you get aggressive cancers. And so the model that was sort of posited was that progranulin normally promotes cell growth and survival. And so if you don't have enough progranulin, then you have less cell growth and survival, and you get neurodegeneration. Okay? There's just one problem with this model, which is that people couldn't show that it was true. All right? So people didn't know, still didn't know, what is progranulin really doing, in particular in the brain, and how does not having enough of it lead to neurodegenerative disease. And so that was when Bruce suggested to me, hey, maybe you should look at this in worms, Amy. And so I decided to do that. So the thing about worms, the thing you can do to worms that obviously we can't do to humans, is we can mutagenize them. And then you can knock down certain genes and look and see what happens when you knock down these certain genes. And believe it or not, there are two groups in the world, one group in Japan and one group in Oklahoma, where it's a full-time job for them just to knock down different genes in C. elegans. And this group in Japan had actually done this. They'd mutagenized the worms. They'd looked at the worm babies, and they found a worm baby that was missing progranulin. And they put this on their website, and I said, oh, that's what I need. I asked them if they could send it to me. I got it. So I got this worm. It has no progranulin. Okay, so what does that look like? What does a worm with no progranulin look like? It looks totally normal. And when I say normal, it's normal size. It grows at about the same rate as a worm with progranulin. It moves around just fine. But that's okay. I actually wasn't too upset about that. We looked at a couple of other things. We looked at the lifespan, and this is something you can do very easily in worms, and you can do not very easily in flies, and even less easily in mice. And it's just impossible to do in humans. You can do a full lifespan. So I looked at worms with normal amounts of progranulin, with no progranulin, or with half the normal amounts of progranulin. And we start with, let's say, 100 worms, and then we can follow the fraction alive over time. And this is days. This is the average lifespan of a worm is about 18 days, okay? And if you compare the blue line, which is normal, to the animals that are missing progranulin completely, which is red, they're superimposed on one another. So the lifespan of these progranulin mutant worms, these worms missing progranulin, was also normal. Just to show you there was a difference, we can measure how many total progeny these animals have, and each animal has about 300 progeny, which is great if you want to amplify very quickly. So there was a slight difference in the number of progeny. Okay, so we see a subtle difference. So I actually wasn't upset when we found this, because people with low levels of progranulin actually do very well until late in life, because this is a neurodegenerative disease that doesn't present until the 40s, 50s, 60s, or 70s. So it just meant we needed to look a little bit closer. And so we decided to do that, and specifically we decided to look at the process of programmed cell death, okay? And, oh, this was a slide that was to remind me to say the big question is, what is progranulin doing, and how does it cause neurodegenerative disease? So we looked at programmed cell death, okay? So what really is programmed cell death? Well, I like to think of it using tadpoles, okay? I have 2 kids, and we do nothing but watch, these nature shows all day. We just watched a show yesterday about tadpoles. And so tadpoles, as we all know, are laid as eggs. They hatch. They swim around for a while. At some point, they grow some hind legs, and then they grow some forelegs, and then they turn into this thing that looks pretty much like a frog, which is down here, except for it has this tail, okay? Now this frog, this tadpole frog, needs to get rid of this tail, because it would just never be accepted by the regular frogs if it had a tail, right? So how does it get rid of that tail? And the answer is programmed cell death, okay? Somehow the cells in that tail know it is time to die. And it's still somewhat of a mystery, but it's the process of programmed cell death. It's also called apoptosis. That is, it's built into the development of from the tadpole to the frog, that at a very specific time during this animal's development, those cells in the tail just decide it's time for me to die, and the tail falls off. The same thing happens in human embryos, because as we develop as embryos, we actually develop initially with webs between our fingers, and that webbing, just like ducks, and that webbing actually has to remove itself. And so it's the process of programmed cell death that tells those cells in the webbing it's time to die. This organism doesn't need you anymore, okay? So programmed cell death occurs when cells are no longer needed, and sometimes it occurs when cells are damaged and they need or abnormal. And so cells can say to themselves, I'm not normal, I need to die. And that process is programmed cell death. Okay. So programmed cell death is a fundamental biological process. And it's also a complex process, as you can see here. But that's the actual details of this are not important. What is important is that it's very highly conserved. And I told you already that a lot of these details were worked out in C. elegans. And so we know that there's a strong analogy between the molecules that control programmed cell death in worms compared to humans. And this work was actually made possible by the work of John Sulston. So what he did is he took, this is an egg, a C. elegans egg, and this is a single cell inside this little eggshell, okay? And so John Sulston actually literally sat at the microscope all day, and he watched these single cells divide into 2 cells, and he gave those 2 cells names, and then 2 cells divided into 4 cells, and he gave those 4 cells names, and then he watched all of these cells divide, and he drew a lineage of all the cells. And this is one page from the lineage. There's actually 13 pages of these cells, and he gave them names that are really interesting, like ABLLPLARP, okay? But he was able to follow every cell, and because there are only 960 cells in a full-grown C. elegans, he was able to map out every single cell. And as he did that, he noticed that some cells actually take on this weird morphology, and then they disappear. And those are the cells that die. And those are some of these X's right here, okay? Now, as time has passed, we have more sophisticated ways of looking at programmed cell death, and we can measure the time a cell is born to the time it starts to undergo the process of programmed cell death, and that's called the time to die. And then from the time it starts to show the morphological features of dying until the time it disappears is called the time to clearance. And I'm just going to tell you the punchline, which is these animals with no progranulin have an abnormality in the time to clearance. And this is how it's illustrated. So this is an older egg. It has lots of cells now. And here are 3 cells that are dying, okay? Cells that are undergoing apoptosis or programmed cell death. And they have this sort of process of maturing. And when you have normal levels of progranulin, the process of maturing goes, you get this nice flat, we call them refractile discs or buttons, and they mature until they disappear, okay? And it takes about an average of 10 minutes in a normal C. elegans. If you don't have any progranulin, you go through the same process, the same sort of maturation of these refractile discs, but it only takes half the time now. So if you don't have any progranulin, the time to clearance of these refractile bodies is half the time it normally is, which means that the normal function of progranulin is to slow the clearance of dying cells. Okay? So without progranulin, this process of dying cell clearance happens much more quickly. So when there is progranulin, it means progranulin is saying, slow down, don't die so fast. Okay? So I like to have a little cartoon. So this is a cell. It's decided to die. All right, so it's sad. What I haven't mentioned till now is that this dying cell gets a little bit of help along the way. It actually can't do everything on its own. There's actually usually a partner, and usually it's this cell's sister in C. elegans, and I'm going to call this cell the engulfing cell. So the engulfing cell actually comes along and helps the dying cell. It actually eats it up and helps digest it. Okay? So I just told you, with no progranulin, this process of sort of engulfing and being eaten happens much more quickly. So we were sort of wondering, does progranulin act on the engulfing cell or the dying cell or both? Okay? And I can tell you that we think it acts on the dying cell, but we're still in the process of trying to figure out exactly how. But we have shown that the engulfing cell is affected by progranulin. And the way we did that was to move from worms to mice. And some colleagues of ours at the Gladstone Institute here at UCSF, Bob Fries in his lab, had made a mouse that doesn't have progranulin. So we got some engulfing cells, some macrophages from those mice, and we looked to see how long does it take for it to eat things, okay? So this is in a macrophage with normal levels of progranulin. This is a macrophage here, and here's another one here. And we just fed it some glowing beads, some fluorescent beads, and we looked to see how many can a macrophage with normal amounts of progranulin eat in a certain amount of time, in 90 minutes. And this one has 2, 1, 2. This one has 1, 2, 3, 4, 5. So, you know, an average of about 3.5, okay, beads in 90 minutes. Well, then we looked at some macrophages that don't have any progranulin. And lo and behold, they eat a lot more beads, and they eat them faster. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 11, okay? So basically, we've gone and shown a process is perturbed in worms, and then we have taken this finding and translated it into mice and shown that mouse macrophages also have an abnormality in this process, meaning if there's no progranulin, mouse macrophages can engulf faster. So, keeping in mind that not enough progranulin causes neurodegeneration and too much progranulin causes tumor growth and metastases, based on these findings, we then developed a model for how we think not having enough progranulin can actually cause neurodegeneration. And the model goes like this. So let's say we have normal levels of progranulin, and then we have a healthy cell, okay? So let's give this cell a little bit of damage, and we'll say it's non-lethal damage, okay? And so this cell becomes sick. But because by definition this damage is non-lethal, we would expect the cell to recover, okay? And only if something else is wrong, or very rarely, would this non-lethally damaged sick cell have to die. Normally, the cell would recover, okay? But let's say we don't have enough progranulin. Now this sick cell, instead of recovering, instead it is eaten or engulfed prematurely by these very sort of aggressive engulfing cells. And you could imagine if this slightly damaged cell was a neuron, over the course of an entire lifetime, then we could have the development of a neurodegenerative disease. Let's say we have too much progranulin, though, okay? And let's say the thing that's producing too much progranulin is this tumor cell. Well, now this tumor cell has basically thrown up an invisibility shield to engulfing cells. Even though a tumor cell is abnormal, and the immune system would normally say, you're an abnormal cell, I'm going to eat you up and remove you. Now this tumor cell is pumping out progranulin, which is slowing down the ability of an engulfing cell to eat it. And so now the tumor becomes resistant to engulfment, and the tumor can grow and be very aggressive and metastasize. So that's our model of how progranulin can cause problems when there's not enough and when there's too much, okay? I should say, as a caveat, that this is still a model, and there's lots of work that needs to be done, okay? But now we've gone from PIC describing frontotemporal lobar degeneration, organizing our cohorts, identifying progranulin, and now we've found that progranulin deficiency accelerates the engulfment of dying cells, and now we have a new target, which is programmed cell death, the interaction between the immune system, the engulfing cells, and neurons. But we want to get over here. We want to develop some treatments. And I'll say that this work that I just talked about was literally published, I think, 3 weeks ago. Okay, so this is new stuff. So we're not here yet to diseases and treatments. But I'm lucky enough that I work with a group of other scientists, both here and at other institutions, called the Consortium for Frontotemporal Dementia, as part of a Bluefield project to cure FTLD. Okay, and other labs in this group have been really pushing the frontiers of trying to find treatments for this disease. I'm going to tell you briefly about 2 of these stories. Okay. Oh yeah. So an explanation for what they're doing. So I mentioned to you already that the progranulin gene is on chromosome 17. In people who have the disease because of a mutation in the progranulin gene, usually there's only one copy of the gene that's abnormal. So there's one abnormal copy, it's a progranulin gene with a mutation, and no progranulin protein is produced from this gene, okay? But there's still a normal progranulin gene, and this gene is producing normal protein. And for fairly complicated reasons, the levels of progranulin are regulated pretty tightly, and so the body normally would not produce more than half of what we really need in terms of total amounts of progranulin. But the idea that these other labs had was maybe we can boost the production of progranulin from this normal gene. So the labs of Joachim Hertz and Gang Yu at UT Southwestern started to just look at drugs that were already on the market, so FDA-approved drugs, and to look to see, can we boost the production of progranulin with any of these drugs? And they screened through tens of thousands of compounds. And this is progranulin that's on what's called a Western blot. So it's a way to image the amount of progranulin that's being produced by a cell, okay, or cells in this case. And if we look at just compound 1 here, there's no compound 1 here, so we have just a little bit of progranulin. But with increasing amounts of compound 1, we get increasing amounts of progranulin. That's great. And they showed that if you add compound 2, you could get even a little bit more. And as it turns out, compound 1, and this work was literally published yesterday, is something called SAHA. Okay? It's a chemotherapeutic agent that is currently in use. Okay? It does have some toxicity, so it's probably not something that someone could take every day to boost their levels of progranulin. But it's in a family of chemicals called HDAC inhibitors, or histone deacetylase inhibitors. And you might have heard of this because resveratrol, which is the chemical that's thought to cause red wine to be good for you and improve your health span, is also a histone deacetylase, or HDAC inhibitor. Okay? So this is pretty exciting. And so here's just, you know, you have Gong and Jakob had this idea. We can just look for compounds that are already FDA approved and look and see if they increase progranulin. And lo and behold, they did. They also did this really neat thing where they got these mice, which they can image in a way, so basically the mice glow in the areas that they're producing progranulin. And so this is a progranulin, this is a mouse that's producing normal levels of progranulin. And then they feed this mouse SAHA, this compound 1, and you can see there's a lot more. So the redder the color, the more progranulin is being produced. Okay, so that's just sort of a space-age picture of a mouse. Okay, so then Lee Gann here at UCSF at the Gladstone Institute also thought, she actually approached this a slightly different way, but she also thought, let's look at certain compounds to see if we can boost the amount of progranulin that's produced. And she also found a compound which, when she fed it to mice and then looked at the levels of progranulin in the brain, she actually found that she could almost double the amount of progranulin produced. So that's also very exciting. And this work is not yet published. Okay. So that's the frontiers in FTLD research from worms to mice. Now let me tell you just another short story about ALS research. And this will take us from yeast to flies all the way up to humans. Okay, so this story, I'm backing up now. We're back at the genes again. Okay, so now we're at TDP. We're going to talk a little bit about TDP43. So what is TDP43? So TDP43 is a sticky protein that can make cells sick. That's the bottom line. Okay. It was actually interesting how this protein was identified, though, because actually a group at the University of Pennsylvania decided to look for, they actually looked in these families of people who had progranulin mutations, and they knew that there was some sticky protein in their brain. And they actually went through and purified that protein and found that it was TDP43. Once that protein was identified, then people started to sequence the TDP43 gene to actually look and see if there were any mutations in the gene. And then they discovered that mutations in TDP43 cause ALS, or Lou Gehrig's disease. And so the question is, what makes TDP43 sticky? Now, what makes it bad for cells? Because it's actually a protein that it's involved in processing RNA. That's not that important. But the important thing is, it's sticky. And why is it sticky? And what makes it more sticky to the point that it no longer can do its normal function and becomes bad for neurons? Okay? So another group at the University of Pennsylvania decided to try and ask this question using yeast. And what they did was they engineered yeast to produce TDP43. So this is a little schematic of a little pink yeast producing sticky TDP43. Okay? And they compared the yeast that produced TDP43 to normal yeast. Okay? So here's a group of normal yeast. Okay? And they're growing, they're happy, they're eager to make bread. But then we give them some TDP43, and they don't grow as well. Okay? Because they've got the sticky protein gumming up their insides. So this was the initial observation. If you make these yeast produce TDP43, and this is human TDP43, the yeast don't grow as well. So what this group, and this was work done by Aaron Gittler, they wanted to figure out what other things can we do to yeast that would make the TDP43 even more toxic. Okay, so what can basically enhance the toxicity of TDP43? And because this is yeast, yeast have about 10,000 genes, they literally went through and they tested every single gene in the yeast genome to see which ones make TDP-43 more toxic, specifically making TDP-43 more toxic. And they do these assays where they grow up the yeast in what are little colonies. And this is just a schematic, because I'm going to show you some actual data on the next slide of a colony that grows really well, of a colony that grows so-so, and a colony that's not growing very well at all. Okay, so these are colonies of yeast on little agarose plates. So here's what they found. These are control yeast, and this is what's called a dilution series. So I'm making up these numbers, but let's say this is 10,000 yeast, 10,000 yeast to start, 7,500 yeast, 5,000 yeast, and 2,500 yeast. Okay, and so you can see that even though these grow very well, there's this nice pink color. There are not quite as many yeast here as here. Okay, and these are the control condition. These are the normal healthy yeast. Like I said, if you make these yeast produce TDP-43, they don't grow as well. And you can see by the time you get down to this 2,500 yeast, barely any are growing. So what they actually found is that this gene, ataxin-2, which is actually called something else in yeast, but the human gene is ataxin-2. Normally, ataxin-2, if you produce it in the if you produce extra ataxin-2 in the yeast are fine. But if you have extra ataxin-2 and TDP-43, then the yeast grow even worse than they do with TDP-43 alone. And I don't know how well this is projecting, but you could tell this is a little pinker than this. This is a pretty white colony. Okay? So their finding was that yeast ataxin-2 causes TDP-43 to be more toxic. All right. That's a pretty neat finding. But this is science. This is the real world. And we actually find a lot of things that aren't true. So I'm pretty sure no one went running down the hallway shouting that they'd found the cure to ALS and TDP-43. They needed to test it because this is yeast, okay? So we can't go to humans and test it. That's not a possibility. They probably found a lot of things. So it's not actually even practical to test this in mice because that's very expensive. So what they decided to do was to go to flies, all right? They decided to test this interaction to see if it was true in flies as well. And so this is a picture of the Drosophila, giant Drosophila. And one of the nice things about Drosophila, I actually don't like Drosophila. I think they're really gross. And I work next to a fly lab, and they're always like dive bombing into my tea and stuff like that, so I actually don't like flies. But they are very useful because they have these big eyes, okay, these big old eyes. And the eyes are part of the nervous system. So if we want to test something in neurons in the nervous system, we can look at their eyes. So this is a close-up of an eye. I shouldn't say that. I feel really bad that I said I don't like flies because they're incredibly useful. But because I like bananas, and you know, your banana gets a little brown, and then there's just flies like literally buzzing around your head. But okay, so this is the fly eye, and this is a normal fly eye. Okay, and there are these things called ommatidia. And if you look at this fly eye in cross-section, you see this beautiful organization of the fly eye. Okay, so this is what's normal. If you put TDP-43 into these flies, this eye looks a little, it's called rough. It looks a little rough. And down here, you can see that this normal organization is lost. So in this side view, you can see how this eye really doesn't, it's not in good shape. Now, if you put ataxin-2 into the eye actually looks fine. But if you put TDP-43 and ataxin-2 in, you can see this eye is a mess, okay? You can see it even here. It's just a sick-looking eye. So basically, they've made this finding in yeast. They've confirmed it in flies. Now, here's the big question. Is it going to hold up in humans? Okay, so ataxin-2 worsens TDP-43-induced eye degeneration. So I'm going to tell you just a little bit about ataxin-2. So ataxin-2 is this really interesting gene that was already associated with a neurodegenerative disease called spinocerebellar ataxia type 2, or SCA2. And it's one of these, it's called a polyglutamine disease or a CAG expansion disease. Because in some of our genes, there are these regions where we have CAG, 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 CAG. And when those are nucleotides, and when that translates into a protein, you get glutamine, 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 glutamine. And you might have heard about this in Huntington's disease. So Huntington's disease is a polyglutamine expansion disease. There's too many CAGs. Same thing in Fragile X. It's a triplet repeat disease, okay? And so what was already known is that in the ataxin-2 gene, if you have greater than 33 of these repeats, you will develop ataxia. You'll get the spinocerebellar ataxia. But it's sort of thought that that was the cutoff. In less than 33, that there was no known risk. So what Aaron Gittler and his colleagues did were they went and they found a bunch of individuals with ALS, not with spinocerebellar ataxia, with ALS, and they sequenced the ataxin-2 gene. And what they found was that, indeed, okay, we already knew, greater than 33 repeats, you get ataxia. But they actually found that if you have between 24 and 33 repeats, you're at increased risk for ALS. And this was a pretty robust association. It looked quite real. And now the sort of normal level of CAG repeats is less than 24, where you have no known risk. So this is a great story. They start with yeast, baker's yeast, and they find an interaction. They show that it's true in flies, and then they find this entirely novel interaction now in humans. And so if we go back to our schematic here, now they've shown that expansion of ataxin-3 causes TDP-43 to be more sticky, and so you have this new target, this interaction between TDP-43 and ataxin that could potentially be targeted for drug interactions. Okay, so that's sort of my story. As a quick summary of this part, I hope I've shown you that the study of human diseases relies on contributions from multiple disciplines, and that model organisms such as yeast, worms, and flies have been and will continue to be very important in understanding neurodegenerative diseases and in developing therapies. And finally, I think it's important to mention that the long-term benefits of research endeavors just can't be predicted. And so even though I've talked mostly about what's called sort of translational research, research that's targeted at trying to treat human diseases, it's also really important to support basic science research with model organisms, because you just don't know where that basic science research is going to lead you. And by basic science, I mean just trying to understand fundamental biological processes that we don't know that are necessarily related to any diseases. And I think the poster child for this idea is this little fella, Aquaria victoria. I don't know how many people have been down to Monterey and seen the jellies there, but this is the little glowing jellyfish, okay? And so some marine biologists were out there studying the jellyfish. They noticed that if you shined a certain wavelength light on these guys, they would glow. And then through that, the gene that was responsible for the protein that made these guys glow was identified. And from that, we got GFP. And from that, Martin Chalfie won a Nobel Prize, and Roger Sand won a Nobel Prize. And so that's basic research that has now really transformed the landscape of our ability to look at processes, living processes. And so I just like to put in a plug for basic science research as well. And that's it. Thanks.
So today I'm going to be talking about seizure of sticky proteins in stem cells. And I know the topic sounds a little bit disjointed, but hopefully by the end of this talk you'll understand a little bit about the connectedness that we're studying, and particularly in the mouse model. I'm going to extend upon what Dr. Amy Gao has told you about in her model organisms by talking about the mouse model, and as particularly the mouse model of Alzheimer's disease. I'm going to tell you about the hidden seizures that we found in the mouse model, which was quite surprising. It's really changed the way we think about the way the brain works in Alzheimer's disease. And then I'm going to tell you about a treatment intervention that I'm very excited about, which is reducing the tau protein and how that may actually be helpful in these Alzheimer's disease models. And finally, in keeping with the theme of today's presentation, I'm going to be talking about neural stem cells, which really I think is a true frontier in neuroscience research. I believe you've seen this slide before, and the reason I show it is just to illustrate the complexity of the disease that we're studying. Alzheimer's disease, there's no doubt about it, is really a complex disease involving a lot of factors, including amyloid beta, which can exist in plaques or in the oligomeric soluble form, tau, which is a cytoskeletal protein within the cells and can aggregate in the disease, apolipoprotein E, which is the strongest genetic susceptibility gene that we have found to date. The vasculature can have an effect on this whole system, as well as the inflammatory system. And so obviously it is a complicated disease, but in order to really try to investigate such a complicated disease, we're using model organisms to try to simplify and reduce the amount of molecules that we're looking at one time. And we're really trying to focus in on the molecules that we think are really vital in initiating the disease. And I've chosen to study mice basically because it is a good model organism for the questions that I ask. When one considers and looks at different model organisms, there are a lot of criteria that we think about that are really essential criteria for a good model organism. One is that they have a relatively short lifespan. I know mice have longer lifespan than flies or than yeast, but the relatively short lifespan allows us to look at the disease process at early stages and then as the mouse ages. As well, the entire mouse genome has been mapped, and so we know every gene that a mouse has. And it's really surprising that there are a lot of, basically almost all of the mouse genes that have been mapped have a related human gene. We have transgenic mouse models, just like with the flies and the yeast, and with the worms, we have transgenic mouse models that help us isolate the effects of single genes. And we can look at the effects of these genes in the mouse models one at a time. One example is the APP gene. This is a gene that is making the amyloid precursor protein, and that is a gene that has a mutation in some families that are linked to the disease. This particular mutation, the APP gene, which is the amyloid precursor protein, causes the amyloid precursor protein to be cleaved at a particular site that produces a lot of these toxic amyloid beta fragments. We can introduce this gene, this human gene, into a mouse, and then this pregnant mouse's progeny will carry, some of them will carry the gene. There are many relevant functional measures in mice that we can look at. These include behavior and cognition, and we have a lot of sophisticated tests of memory and learning in the mice that we can do, as well as looking at things like intracellular trafficking of molecules and the electrical activity in the brain. We also have a lot of biomarkers, biological markers that have been seen in the human condition that we can see in the mice. And then we can also look at the mouse model and verify whether what we see in the mouse happens in humans. There are a lot of examples of this. There are even examples of proteins that were discovered in my lab that I work in Leonard Mucke's lab that were first discovered in the mouse model and later actually identified in humans. Now, a mouse brain is clearly not the same size as a human brain, but despite the size differences, there are a lot of biological similarities. Mice never develop Alzheimer's disease spontaneously, but when the mouse brain is exposed to this amyloid precursor protein, especially with familial mutations, it develops a lot of amyloid beta in the brain, and this amyloid beta, which you've heard about in previous talks, can aggregate together. It's a sticky protein that aggregates together and can form these plaques, which are shown here in red. And these plaques can engulf passing neurites, neural processes from cells. And these plaques look pretty nasty. They look like they could really have a bad effect on the brain. And up until recently, we actually thought that the plaques and actually the tau pathology was the culprit, was really causing the problems in the brain. But we're learning actually from these mouse models and other model organisms that it may be actually soluble species of these proteins that are causing effects, toxic effects on synapses and neurons before they even deposit. And these depositions may actually be a protective mechanism. And so actually, we can't see these soluble forms of the protein very well. And so we turn towards functional measures to really get an idea of what the effects of these toxic proteins are functionally on, for instance, memory and learning. One of the most powerful tests for learning and memory that we have is the Morris-Water maze, named after Richard Morris, who developed this test over 30 years ago. This is a task where I think you saw this video from Kate Posen as well. We are placing a mouse in a tub of opaque water, and the mouse is asked to find this hidden platform where it can escape from the water. And it has visual cues around the room, like crosses and X's, of different colors, and it can figure out where the platform is in relation to the objects or the drawings around the room. And over time, these mice learn quite effectively where that hidden platform is. When mice have a human APP gene and these high levels of amyloid beta in the brain, they are no longer able to find this platform as well. And you can see this mouse is really searching and not really knowing, he really doesn't have a clue where the platform is. We've all experienced similar scenarios, looking for our car or looking for our keys, which is not necessarily pathological. But if it happens every day, that's when we began to worry about these mice. And so now that I've introduced the model, I'm going to tell you about a surprising finding that we discovered in our lab that has really changed the way we think about the way the disease progresses and how neurons communicate. We know that neurons communicate through electrical impulses. And so you've seen slides where neurons are communicating with each other. These neurons communicate by electrical impulses that are initiated from neighboring neurons, and then this neuron is activated from a neighboring neuron through its dendrite, and then that causes this particular neuron to propagate to generate its own electrical signal, which propagates down an axon to distal axons along that same circuit. And so normal brain activity, if you think about normal brain activity, you'll have a neuron that fires within a circuit, and then it activates the other neurons within that circuit in a ordered and purposeful fashion. We can actually record brain waves through EEG or electroencephalograms, and the brain waves for normal brains has this very smooth appearance and with a defined frequency that we always expect to see a certain frequency, and we expect to see these smooth waves from a brain that is behaving normally. From a brain in a patient with epilepsy, we often see epileptic activity. And so if you think of neurons communicating as chatter, and you think of one analogy would be like you're in a large restaurant, and it's a crowded restaurant, and all the time there's low level of chatter around the room. That's normal. And even though there's a lot of chatter and people communicating with each other, you can still communicate with the others at your table. So every table should respect every other table, and so conversations are able to be held normally. However, if you're, for instance, like in a pub, and there's a soccer match on, and all of a sudden the striker scores a goal, and his name is Bruce, let's say, and the crowd loves Bruce. He's just a great scorer, and so as soon as he scores, the crowd goes, "Bruce, yes!" And no one else is really able to talk because they keep yelling, "Bruce, Bruce, Bruce." And this keeps going on, and no one can really understand what they're saying to each other. Every time the neurons fire together, this is an epileptic discharge. Epileptic discharges on an EEG have this really sharp appearance. And we call it a spike because it looks like it would hurt if you sat on it. That's our general rule for what is a spike. Now, a spike can occur in this pattern like this, and it can actually interrupt the communication of the other neurons. And often a spike is followed by a slow wave, which just shows that the brain is not behaving normally. What we found in our mice was that if we looked at this protein that is expressed after it is activated, this activity-regulated protein ARC, in our normal mice, which are called non-transgenic mice, we saw a low level of chatter, which is expected. A few neurons are very active, some are somewhat active. In our APP Alzheimer's disease mice, we saw that just a few neurons were communicating properly in most of the mice. But then in a certain subset of mice, when we took a snapshot of the brain, we actually saw that there was this massive overexpression of the ARC protein. And this was really surprising to us and made us think about the fluctuations in the brain activity that may be going on in these mice. And it led us to this hypothetical model where in the normal mouse you have generally low levels of fluctuation in the activity over a day. And then in the APP Alzheimer's mouse we have intermittent little bursts of brain activity followed by more prolonged suppression of brain activity. And we think that these bursts of activity cause some remodeling in the brain that actually is compensatory and tries to clamp down on that. And that's why we actually see prolonged periods of depressed activity. And so in order to look at the brain activity in the mice, we have actually monitored these mice by EEG, brainwave study, where we can actually look at mice that are freely moving and they are attached to an electroencephalogram. So what I'm showing you here are normal mice in an Alzheimer's disease mouse model. And so this normal mouse is in this cage, in this apparatus here, and then this Alzheimer's mouse is in this one here. And this normal non-transgenic mouse, the recording from this wire is represented by these gray tracings, and they look pretty smooth. This is kind of squished, so they look a little bit sharp, but this is normal. And then the Alzheimer's mouse brainwave are shown here, and you can see we're capturing spikes that are occurring in this Alzheimer's mouse brain. I'm going to show you a video of how these spikes can actually amplify and become more rhythmic. And when that happens, we call it an electrographic seizure, because the brain is constantly having these repetitive firing that is really taking over the brain and not allowing any of the rest of the brain to do its normal functions. During this seizure, the mouse is just frozen in space. It's not doing anything. And so we call this a non-convulsive seizure. And we also, we all know about seizures, and I think a lot of people think about seizures as these grand mal, drop to the ground, convulsive seizures, but there are other types of seizures that are less overt, and they can be such things as absence spells or staring spells that you can see especially in children, or partial seizures that can just impair one part of the brain without affecting the entire brain. Actually, what's amazing is that after the seizure, the mouse is just moving around just like it did before. And so if you just looked at the mouse without recording its brainwaves, you wouldn't really even suspect it was having a seizure. And so from these mouse models, we have really looked at the effects of amyloid beta at different levels of complexity. And what we have seen is that amyloid beta can have different effects on the brain in different levels of complexity. Amyloid beta seems to suppress activity at individual synapses and neurons, and then it causes an imbalance of hyperactive and hypoactive circuits. And because of that, there's a synchrony that happens, which can lead to epileptiform activity at the network level. So after we discovered this in the mice, we were very curious about what is the clinical relevance. We know that epileptic seizures can occur in several mouse models, but and that's been shown in several labs, but what is the incidence of non-convulsive epileptiform activity in Alzheimer's disease? And so actually we do know that there is some overlap between epilepsy and Alzheimer's disease. It turns out that up to 10 to 20% of patients with the condition will have a convulsive seizure at some point in the disease. And we've often thought of that as a consequence of basically end-stage disease. So people who are really in the severe stages, because the brain has lost a lot of neurons and has scarring, that's why the seizures are occurring. But when we look at epidemiological studies, we find that the seizures can occur actually at any point in the disease course. And since we found these seizures in the mice, we're really, I think, a little more vigilant about seeing them in our patients. And we've come across a lot more patients that seem to have epilepsy and epileptiform activity on their EEG. We also know that EEG abnormalities and seizures are much more common in familial cases where there's an overproduction of APP, especially with genetic mutations, and in Down syndrome, or trisomy 21. So Down syndrome is caused by an extra copy of chromosome 21, and it turns out that the APP gene is on chromosome 21. And so Down syndrome patients, or Down syndrome people have a lot of APP in their brains, and some of them, actually a lot of them, later in life do develop Alzheimer's disease in their 50s, and they also, a lot of them develop seizures. We also know that epileptic discharges themselves can impair cognition. Epileptic discharges, if they're occurring during a neuropsychological test, can actually impair one's ability to do a specific task. And it seems localization related. So if somebody's having left temporal epileptic discharges when they're asked to remember a word, they may actually not be able to remember the word because the left temporal lobe is important for verbal memory. And so we were really curious about what is the extent of subclinical epileptiform activity in the human condition. And the reason we're curious about this is because we already have seizure medications that could potentially benefit these patients that have epileptiform activity, especially if the seizures are having an effect on their thinking abilities. And so I've launched a clinical trial with folks here at UCSF, and we're enrolling people to answer that very question. We're enrolling with people with mild cognitive impairment, Alzheimer's disease, dementia with Lewy bodies, which is the Parkinsonian dementia, frontotemporal lobar dementia, degeneration, which you've heard about, and normal controls. And we enroll these patients actually in the hospital for 36 hours of EEG monitoring. And so I really am grateful for these volunteers who are enrolled in this study. And we place these electrodes on their scalp in this way. This is their nose, this is their ears, and you can see how the electrodes are placed on the scalp. We have some additional electrodes attached to the parietal region, which is a region of interest. And we also do a study which is called a magnetoencephalography, or it's like a fancy MRI study that looks at magnetic wave activity. We can actually see epileptic activity through that means as well. We're looking at clinical fluctuation scores and other imaging and genetic and neuropsychological tests which we can use to correlate with any findings of epileptic activity, so we can know the real functional consequence of these epileptic discharges. So far, we've enrolled a few subjects, and we're starting to pick up some epileptiform activity. This is not published and really in the preliminary stages. So this may or may not hold true, but we know that seizures do occur in humans with Alzheimer's disease about 15% of the time, the convulsive seizures. And we're actually picking up subclinical spikes in about 30% of our subjects who have never had a seizure before. And so this is making us wonder whether there could be this subpopulation of patients with Alzheimer's disease who have these epileptic spikes but have not had seizures. When we look at individual cases, we've actually come across some really interesting findings. This is a 53-year-old woman who developed aphasia, or trouble with speech production. In some ways, she looked like she had had a stroke, but this was actually something that had occurred insidiously and was progressive, and she had trouble with her speech output. She had a PET scan which showed amyloid deposition in the brain, and an FDG, or glucose PET scan, that showed reduced glucose metabolism in the left frontal lobe and the bilateral temporal parietal regions. This pattern is very consistent with Alzheimer's disease. And of course, as we see the amyloid deposition, this really confirms the diagnosis that we had suspected. She had frequent epileptiform spikes in her left frontal lobe, which is an area that is very important for speech production. And so this was a really provocative and interesting case. And so we're really wondering about whether these epileptic spikes could have a functional consequence. And now I want to tell you about basically switching gears just a little bit and looking at the mouse models again, because we're having to turn back to the mouse model to think about what kind of treatments we could offer for a patient that's having these epileptiform discharges. We have a lot of FDA-approved anti-seizure medications, but some of them have cognitive side effects which limit their use in cognitively impaired population. And so what we focused on is, in our lab, is the microtubule-associated protein tau. And the reason for that is that we know that amyloid beta has bad effects on the brain, but the amyloid beta plaques, like I said, do not seem to correlate very well with disease severity. As you have heard from, I believe, Gil Rabinovich, well, before I say that, we're looking for mediators of toxicity. Amyloid beta can have effects on their own, but we're looking for mediators by which A beta may impair neuronal and cognitive functions. And so we were interested in the tau protein because actually the tau protein seems to correlate better with disease severity. The aggregates of the tau protein, where they're located and how they progress, seem to correlate better with the disease progression in the temporal lobes and then spreading to the cortex than amyloid beta. So tau is a protein that is actually pretty simple in its structure. It has this native unfolded structure, and it sits on the microtubule, which is a track and a cytoskeletal a long cytoskeletal sort of tube that keeps the integrity of the cell intact, and it also serves as a track for transport of cargoes. Tau can normally reside on the microtubule, but it can get phosphorylated. That's these little phosphate groups shown with P's. And then when it's phosphorylated, sometimes tau detaches from the microtubules. Not all of the tau, but then the tau that detaches can take on different conformations. And then tau, like A beta, like TDP43, is a sticky protein, sticks to other tau proteins, and can form these soluble oligomers. These oligomers themselves could be toxic to the microtubule, and they may also form these filaments, which we see under the microscope, these neurofibrillary tangles. And we're really curious about what tau is doing and where is it toxic. And is tau actually just a bystander, or is it actually having an effect on the cell? And so to answer that question, we took this genetic model and we actually took mice that had the APP gene and crossed them with mice that had the tau gene knocked out. And so now we have mice that are transgenic for APP, but with tau reduced. As you'll remember, in our normal mice, they have no amyloid beta in the brain, and then they're actually quite good at finding where the platform is. This is actually a hidden platform. This is actually an absent platform where we're just looking where they swim after the platform's removed. It's called a probe trial to test their memory retention. This mouse keeps looking for where that platform was. Can't find it, but it keeps looking, and we know that it knows where it is. This mouse initially it basically, like we showed before, has no clue where the platform is. And this is a mouse with the APP mutation. Remarkably, if we actually remove the tau protein completely, the mice are fine, which that in itself is amazing. Tau is a really prevalent protein in our bodies, but the mice seem to do okay without it. And there may be other proteins that take over its functions. But what's also remarkable is that if you remove the tau protein, these mice have normal memories. And their memories are as good as the mice without the APP gene. What's also fascinating is that the tau protein, when you remove it, can actually eliminate the epileptiform activity that we saw in the APP mice. And it really is effective at this. So this is a curious finding, but it really makes us reassured that the epileptiform activity that we suspected was having an effect on cognition may actually be having an effect. And that because this intervention that reduces epileptiform activity improves memory. Because tau is concentrated in the axon, I've been looking at the effects of tau on the transport of cargoes up and down the axon. And the reason for that is because I think that might be a reasonable place where amyloid beta and tau converge in disease pathogenesis. So what is axonal transport? Axonal transport is the shuttling of different cellular cargoes between the cell body and the synapse. Many of these cargoes basically need to be in certain locations, and the cell really coordinates that through axonal transport. Mitochondria are the energy-producing organelles in the cell. They make pretty much all of the energy in the cell, and they are really needed at the synapse, which is a very active part of the cell. These yellow little bars here, these I'm representing as receptors, and so this could be, for instance, a growth factor receptor that needs to receive a growth factor like nerve growth factor from another cell and then bring that back to the cell to keep it healthy. So this is a really important process that's actually impaired in Alzheimer's disease. If you think of the transport like almost like a commuter rail, the microtubules that I showed you can serve as the tracks for axonal transport. The motor proteins, this is kinesin, actually are like the locomotives carrying the cargo along the axon. And this is a motor protein that brings cargoes from the cell body out to the synapse. And this is the way it moves. It almost looks like it's walking along the microtubule. This requires a lot of energy, and it's using ATP as its energy source. Kinesin, as I showed, it looks like this in a cartoon form. And the reason I was interested in tau is that about 10 years ago, a group in Germany, the Mandukov lab, it's a husband-wife team that are really prominent in the tau field. They found that if you overexpress this tau protein, it can have dramatic effects on axonal transport. And they found that tau sort of acts like a traffic cop. When it's bound to the microtubules, it could actually occupy the space that the kinesin would normally want to bind, and it can block kinesin from binding the microtubules. When tau is not around, or if it's in different locations, the motor protein is able to attach and proceed like it normally would want to. And so tau may actually be involved in regulating the detachment and attachment of different cargos. And this may be a strategy that cells figured out to place cargos where they need to go. To look at this question of tau and axonal transport, we've looked at these mouse neurons that are transfected with the green fluorescent protein that was discovered in worms. And so by labeling them with this green fluorescent protein, we can actually find where the axon is. And then we can look within the axon at the mitochondria. The mitochondria here are shown in red. And so the mitochondria are actually moving around quite a bit in our brains all the time. And this axon, as you can see, is very long, and sometimes it's thousands of times longer than just the size of the cell body. So axonal transport of these mitochondria is very important to keep the cell happy and healthy. What we found was that if we track the axonal transport under different conditions, we made some really amazing, I think, observations. One is that when you add this amyloid beta in its soluble, toxic aggregate form, it can actually inhibit axonal transport quite effectively, even within minutes, as you can see here in red. If you add amyloid beta with a scrambled peptide, which is a control peptide that doesn't form these toxic oligomers, it actually does not affect axonal transport, showing that the effect is really specific for the conformation of the A-beta, the sticky A-beta protein. We found that knocking out tau really had not much of an effect on the axonal transport, which was really interesting to see. You can see here by the white bars that reducing tau in half or completely had no effect on the axonal transport. Plus plus means a normal amount of tau. Plus minus means half of the amount of tau. And minus minus means no tau. The plus and minus refer to the alleles. But we did see that tau is necessary for the amyloid beta effects. And so in the neurons that have normal amounts of tau, amyloid beta is really effective at inhibiting transport. But in neurons that have tau reduced in half or completely, amyloid beta is no longer able to exert its effects on the mitochondrial transport. And so if this were happening with things that affect neuronal activity, that could explain how tau protects against the epileptiform activity as well in these mice. And so I've shown you a number of examples of how tau reduction can actually improve cognitive and neuronal function. And one of those is I've shown you that tau reduction can improve memory. Tau reduction can improve the connections between synapses. It can improve axonal transport and epileptiform activity. So we really think that tau reduction potentially could be an effective therapy. We do not know whether reducing tau in the adult brain is a viable option, and so we're actually looking at that right now in the mice. And so this is just to show you that there are a lot of different ways of targeting tau for therapeutics. One is to inhibit phosphorylation, or another is to inhibit their aggregation, or to stabilize their microtubules, and all of these have really received a lot of attention and are actually in clinical trials right now. Some of them look promising. As an alternative, we actually think that reducing tau levels themselves may be protective and could actually be well tolerated, but we need to wait to find that out. And another thing I just wanted to show you, which I think is really neat, is that I'm looking at the mitochondrial movements in the living mouse brain now through confocal microscopy. And this has never been described before, but we can actually see mitochondrial movements in these axons at the surface of the brain. Now finally, in my last couple minutes, I'm going to talk about neural stem cells, which really I think is almost the final frontier for neurology, at least for our generation, and for neuroscience. And so if you think of humans, we all start as one cell, a zygote that divides. It retains its ability to divide and stay immature. But some cells develop new properties, new specialization. And this occurs over several thousands and millions of generations, till eventually we have several different cell types which can form all of the tissues of the body. Some of the cell types, human stem cells, that has received a lot of attention because of their therapeutic and research potential is embryonic stem cells, which come from these embryos, neural stem cells, inducible pluripotent stem cells, which I'll talk about, and adult neuroprogenitor cells. Adult neuroprogenitor cells, I think, are really neat because they're actually occurring in our brains in adults. And actually, we didn't know this until recently. It turns out that adult brains have new cells, new baby cells, that can mature into new neurons. And there are particular centers where this occurs, including the dentate gyrus or the hippocampus. And as you can see in this mouse, there are always a few of these neurons around. Exercise really ramps up the number of stem cells in these mice compared to a sedentary mouse. And these are labeled here in red. And so I use this clearly as motivation for myself and for my patients to really get out there and try to exercise as much as you can. There are a number of therapeutic applications if we could actually replace cells that are dying for neurodegenerative disease. If we could dampen down epileptic activity for epilepsy, if we could replace cells that have died for stroke and also for spinal cord. And also, I'll show you how it's a powerful research tool. We can actually now take cells from patient skin biopsies, and we can derive fibroblasts, which are connective tissue cells, from these skin cells. We can then introduce a lot of reprogramming factors that actually turns these cells into embryonic-like cells. These are pluripotent cells, which means they can turn into really anything. And so these cells were actually formed from an 84-year-old patient initially with motor neuron disease. And these can actually turn back. We can actually reprogram these cells from the skin and make them into neurons. We can actually study that patient's disease in the culture dish. So no, we don't want to clone grandma, but we can look at an individual's cells in the dish without actually having to do anything to that individual other than a quick biopsy. And so we've actually generated dopamine-producing neurons, which may have therapeutic implications for Parkinson's disease, which really affects these cells. And we can make motor neurons, shown here, which may replace the neurons that are lost in patients with ALS. Also, for patients with Parkinson's disease, we think that's a really viable option in the near run, because there are really a subset of neurons that are lost early, and these are dopamine-producing neurons. If we could just replace those that have died in the place where the dopamine is needed, just enough to replete the dopamine that was lost, then that may have a symptomatic effect. And early clinical trials show some promise there. But there are a lot of hurdles we need to overcome. And one of those is that we need to make sure that these cells do not continue to divide, because we've introduced really cancer genes that turn them into immortal cell lines before we differentiate them back into these specialized cells again. And so what I've hoped to impress upon you is that really Dr. Gao and I cover a wide gamut of things, and we've covered a lot of ground today. But as you can see, it takes a real multidisciplinary approach to really think about these diseases. And so in our studies, we've really focused on these model organisms to really try to elucidate mechanisms of disease. And so our area is really right here. And the clinic at the UCSF Memory and Aging Center takes people from here to this point. And so there are a lot of people involved, clinicians and scientists, working together to find targets for these devastating diseases. And we hope that with this multidisciplinary approach that we'll really come up with something effective soon. So thank you for your attention, and we're going to take